First of all, thank you for your interest in this video material. I'm Youngju Ro, who is the CEO of Multilex. I am very glad to have a chance to introduce multiplex technologies in front of you. Multilex technology is for molecular diagnostics. It is about multiplexing PCR technologies, which could be a good platform tech. We called our technology as Megaplex. Our Megaplex is very unique, innovative, and interesting. From now on, I will explain why. Then, let me start. The company, Multilex, has a multiplexing PCR technologies named as Megaplex, and deleted many patents. Our Megaplex is a platform technologies that can have a high value. Multilex was founded with the licensing out of patents as a business model. When you want to make a diagnostic kit, we will be happy to assist you by passing on our skills and know-hows. Here, there is summarized 5 features for our Megaplex system. The first, the Megaplex system is full multiplexing PCR technology, which can detect multi-target and designed as one you want. The second, if you design well, using the Megaplex system, you can detect 7 targets per 1 fluorescence channel. The third, if we have a PCR system having 5 fluorescence channels, you can detect 35 targets by only one single batch. The fourth, the Megaplex system uses a pair of the tail and C-tail. What are they? Please for a while. I will explain later what they are. The fifth, I proudly can say that among the existing multiplexing technologies, the Megaplex system is the simplest and easiest. So from now on, let me introduce the Megaplex system in earnest. Megaplex system is realized using Megaplex probe set, which are probe and complementary probe. I use short C probe. Probe and C probe set include prime and tail respectively. As you can see from the slide, probe consists of forward primer, tail, and fluorophore. C probe consists of reverse primer, C-tail, and quenchers. As you know well, even in the Megaplex probe set, the primers are used in amplifying the target in the PCR process, like other PCR kits. As another essential component of the Megaplex probe set, there are tail and C-tail, which are complementary to each other. And this tail pair plays a very important role in detecting target. I will explain very soon. Primer pairs, tail pairs, fluorophore, and quencher. These are all we need for using Megaplex system. It is very simple, isn't it? So now You may want to know how to detect multi-targets in this megaplex system. Let's take a look at next slide. The slide shows a whole process in megaplex system. There are three main steps. Step 1 is an amplifying step, in which target is amplified by PCR. Step 2 is stabilizing and dissociating step. Probably, you can imagine what will happen between tail and C-tail contained in amplicons. They are stabilized at low temperature and started to dissociate as the temperature rises. Step 3 is an analyzing step. In the step, you can confirm and detect the target by analyzing a fluorescence curve. You need more information? Okay, let me explain it in more detail. Take a look at this slide. In step 1, the probe and the C-probe set are functioned as primer set for PCR. As previously said, the megaplex probe set has a forward primer and a reverse primer, and they each bind to target single strand, which is denatured from double-strand form. This process is the same as the conventional PCR, so you can gain amplified targets, which called as amplicon. So what is the difference? The shape of the amplicon is different because of the tail set. Megaplex amplicons, we can also call it target probe complexes, have a linear-like structures in which tail and C-tail are each connected to both ends of the amplicon as a single-strand form. Don't forget it. At this time, you can see the signal from the fluorescence. It is why the fluorescence is far from the quencher in this amplicon structure. Yes, you cannot see the signal in this time. Take a look at B, tail dissociation step. After A step, we slowly increase the temperature of the tube at the rate of about 1 per minute. In the process, the ring-like shape of the amplicon goes back to the linear-like shape and starts emitting the fluorescence signal again. Do you understand what this means? In the B step, the amplicon shape changes according to the temperature. The temperature is determined by a binding force between the tail and C-tail. Think about it. You can design the tail and C-tail pair for the specific target considering the binding force. And then, if you get a fluorescence graph from them, you can find out the existence of the specific target. Now you can guess the principle for detecting a target in our megaplex system. It is the last step. When the step 2 is completed, we analyze the fluorescence graph in order to get information. Graph A is the example showing the relative fluorescence units which is RFU, during the temperature increase. Graph B is the exemplary graph derived by differentiating the RFU of graph A to the temperature. Then, from now on, let's look at the multiplexing. This slide shows a certain fluorescence graph. You can see two peaks. One is at on T2 temperature and the other is at on T3 temperature. What this means is there are two targets in the sample. In other words, target A corresponding to T2 temperature and target B corresponding to the T3 temperature. Because we designed one set of the tail and seed tail is customized for target A, and this tail set has a binding force corresponding to T2 temperature. And we designed the other set of tail and seed tail is customized for target B. And this tail set has a binding force corresponding to the T3 temperature. This is the basic principle our megaplex can multiplex. Using megaplex system, you can easily and simply detect multi-targets. Just one thing, please consider not to exceed 55 degrees for a label temperature. We believe we will be able to detect 7 targets in one fluorescence channel. Our belief is very reasonable. Take a look at the slide. Only one thing we need is a designing of the 7-degree intervals. Like the table, if tail and seed tail sets will be designed to have label temperatures of 10, 17, 24, 31, 38, 45, 52 degrees respectively, it could be possible. Then, how about 5-degree intervals? In the near future, it will be also possible too. If it does, you will come to detect 10 targets simultaneously per one channel. It is amazing, isn't it? I proudly can say that again. Our megaplex is very unique, innovative, and interesting. If someone wants to develop a kit that can detect 35 targets at once through 5 fluorescence channels, our megaplex will look very attractive. More amazing thing is that our megaplex technology is very easy and simple. However, do not use this technology freely or copy this technology. We have already established a strong patent portfolio. Remember, Multilex megaplex technology is strongly protected by well-managed patents. Take a look at the list on the table. These are patents for megaplex technologies. Multilex has 16 patent applications in 6 countries, including Korea, the United States, Europe, China, Australia, and Canada. 2 of them have been already granted, and another 2 have been allowed in Korea. We can also file divisional applications to remain a potential for patents expanding and optimizing, just in case. Don't worry. In other countries, Multilex will have a patent portfolio. It's similar to the Korea. Then, my explanation is over. Do you think our technology is pretty good? Are you interested? Are you curious? If you want to discuss more with us, please feel free to contact me. You can see the mail address in this slide. I am very happy to wait for your meeting and have a good talk with you. Thank you for your attention.